It's finally the beginning of the end for neurological diseases and disorders. Thanks to scientists at Stanford, we now have a new way to start performing research on certain types of neurological diseases and disorders we were never able to look into before because patients were compromised. But now we can actually isolate the cells and see what's going on behind the scenes. But before we understand what the research is that's making this possible, we should ask who is making this possible. And that is Dr. Sergiu Pasca, a Stanford neuroscientist and psychiatrist who's trying to understand how the human brain develops and what goes wrong in conditions like autism, epilepsy, and rare brain disorders. But here's the big issue he faced early on. You can't just go and take a piece of someone's brain to study it, especially from someone with a psychiatric condition. Unlike cancer or infections where you can look at samples under a microscope, mental health disorders are hidden inside of the brain, and we usually can't study living brain tissue. But don't worry, here come induced pluripotent stem cells. What are they, you ask? Imagine this. You take a tiny sample of someone's skin, like a little scrape of cells from their arm. Then, using a few special signals, kind of like flipping genetic switches, you turn those skin cells back in time, all the way into stem cells. These special cells are called iPSCs, induced pluripotent stem cells. Here's what makes them powerful. They can turn into almost any kind of cell in the human body. Brain, heart, liver, you name it. Now, the process of skin cell into induced pluripotent stem cell might seem a little complex, so let's break it down some more. Think of it like this. Every cell in your body has the same DNA, but different cells use different parts of the DNA to do their job. For example, a skin cell uses genes that make it good at protecting your body, while a brain cell uses different genes. What makes a stem cell special is that it can turn into almost any cell because it's reset to a very early flexible state, like a blank slate. This is all possible because scientists figured out they could take skin cells and reprogram them back into that blank slate stem cell, all through 4 simple steps. First, they introduce a special set of genes called Yamanaka factors into the skin cell. These genes are like master switches that turn off the skin cell program and turn on the stem cell program. Step 2. These master switches include genes named OCT3/4, SOX2, KLF4, and C-MYC. They work together to erase the skin cell's identity. Step 3. Once these switches are turned on inside the skin cell, the cell starts behaving like a stem cell. It can now divide and become other cell types. The last step. Scientists usually do this by using a harmless virus or other delivery methods to carry those genes into the skin cells in a lab dish. That way, they can study these new stem cells. So, in short, you take a skin cell, you infect it with factors that reprogram its gene activity, the cell loses its skin identity and turns into a stem cell, or iPSC, and that iPSC can now be grown and coaxed into becoming brain cells or any other type of cell. So why is this useful for brain research? Because now Dr. Pasca can 1. Take skin cells from someone with, say, autism or a rare brain disorder, 2. Turn them into induced pluripotent stem cells, 3. Then grow actual human brain cells from them in the lab. All of this can be done without touching the patient's brain. Okay, so now that Dr. Pasca has these human brain cells from induced pluripotent stem cells, he goes a step further. He grows them into 3D to form little structures called organoids. Here's where it gets really interesting. These organoids aren't fully brains, but they mimic parts of the brain. Think of them as mini 3D versions of real brain tissue, like growing a mini brain in the lab. So why are organoids so cool? Well, these organoids contain many types of brain cells, like neurons and glial cells, that grow and behave in ways similar to how they do in our actual brains. And the cells in the organoid organize themselves into functional brain-like patterns. like how neurons connect with each other to form circuits. This allows Pasca to study things like how brain circuits develop and what happens when genes go wrong, for example, in diseases like autism or epilepsy. But these organoids aren't enough. While organoids are amazing on their own, Dr. Pasca realized that he could do even more. He wanted to model how different parts of the brain communicate with each other, because the brain isn't just a bunch of separate regions, it's a whole network where different areas work together. So he took things one step further and created what he calls assembloids. What's an assembloid? It's sort of like a combination of different organoids. For example, you might take one organoid that mimics the cortex, the outer layer of the brain responsible for thinking and decision making. Or you might take another organoid that mimics inhibitory neurons, the brain cells that help balance brain activity and prevent it from going haywire. Then you fuse them together into one functional unit, the assembloid. So why are assembloids even cooler than organoids? Because now you're not just looking at one isolated brain region, you're looking at how multiple regions interact. The cells work together like they would in a real brain. And this is important because the assembloid can form brain circuits that help scientists see how neurons communicate and form complex networks. Also, now Pasca can simulate how diseases like autism or epilepsy affect entire circuits. This is huge because many brain disorders don't just affect one part of the brain, they mess with how different regions talk to each other. Here's a real-world example. Dr. Pasca and his team used assembloids to study how pain is processed in the brain. They created a sensory assembloid. It included parts of the brain that are responsible for touch, sensory neurons, the spinal cord, which helps send those signals to the brain, the thalamus, a brain relay station, and the cortex, where pain is processed. They used this assembloid to study a gene called NAV1.7. When this gene is mutated, it can cause pain disorders like congenital insensitivity to pain, where you can't feel pain, or extreme pain sensitivity. Next, they looked into Timothy syndrome, a very rare genetic disorder that affects the heart and brain. Kids with Timothy syndrome often have severe autism symptoms, seizures, developmental delays, and life-threatening heart problems. Timothy syndrome is caused by a mutation in a single gene called CACNA1C that messes with how calcium flows in and out of cells, especially neurons. That calcium flow is super important for how brain cells talk to each other. So how did Pasca study this? Here's the amazing part. First, his team took skin cells from patients with Timothy syndrome. Then they turned them into stem cells, or induced pluripotent stem cells. Next, they grew them into brain assembloids, the mini circuits from the patient's own cells. Finally, they watched how the neurons developed and misfired. Pasca's team was able to see exactly how the disease was affecting brain development and connectivity in real time using human cells outside of the body. This was impossible to do before his technology. But he didn't stop there. Pasca and his team then tested different drugs on the assembloids to find out which ones could fix the problems in the brain circuits. And now, in 2025, they're preparing the first clinical trial for a drug based entirely on what they learned from those brain models, without ever needing to study the patient's brain directly. This is one of the first times a treatment for a psychiatric or developmental disorder is fully based on lab-grown human brain tissue. It's a huge leap in how we might treat neurodevelopmental conditions in the future. So, as a quick recap for what we've seen, Pasca used induced pluripotent stem cells to grow patient-specific brain tissue. He built assembloids to model how brain regions connect. He studied rare conditions like Timothy syndrome using his system. And now his team is launching a real treatment trial based entirely on that lab-grown brain tissue. Here's the best part. Dr. Pasca's lab isn't keeping these powerful techniques a secret. Instead, they're sharing their methods openly with more than 350 labs worldwide. That means scientists everywhere can learn how to grow organoids and assembloids. And they're running immersive week-long training courses at Stanford to teach other researchers hands-on how to build and study these brain models. If you're still wondering why this matters and why it's such a big deal, here's the answer. Because brain diseases affect millions globally, and no single lab can tackle everything alone. By sharing, Pasca helps speed up research worldwide, enables new discoveries about autism, epilepsy, pain, and other brain disorders, and fosters collaboration between engineers, doctors, ethicists, and biologists, combining many fields to tackle the brain's complexity. Pasca credits Stanford's interdisciplinary culture, mixing engineering, medicine, bioengineering, and ethics as key to making this progress possible. Here's the bottom line. Dr. Sergiu Pasca's work is transforming brain science by creating living models of human brain that researchers can study and manipulate. These models help us understand brain development and disorders at a level never possible before. They help us enable new treatments to be tested before they ever go to patients. And they help build a global research community, accelerating discoveries in neuroscience. Thanks for watching. I hope you enjoyed.